Hello, my name is Shelly Krueger. I am the Florida Sea Grant Agent in the Florida Keys for the University of Florida IFAS Extension. And I'm really excited to be talking about marine sponges today, and I thank you in advance for attending this webinar. During today's bite-sized science on marine sponges, we will introduce sponge biology and taxonomy, ecology, human medicine, the economy and sponge fishery, and finally, the threats sponges face and a short review of sponge restoration. When most people think about sponges, they think about the sponges that are used in the bath. And humans have been using bath sponges for more than 3,000 years, which is especially well documented in Greece and the Mediterranean. Yet of the more than 8,000 sponge species, only a couple of dozen are the types that can be dried and made into bath sponges. Sponges range in size from just a few millimeters to giant barrel sponges larger than a human. Sponges grow in many shapes. There are round sponges, finger-shaped sponges, encrusting sponges, and basket sponges, to name just a few. They may be brown and camouflaged, or bright orange, red, purple, yellow, green, or orange. On the bottom left is the giant barrel sponge with a lifespan of 100 years to over 1,000 years old. The photo on the bottom right is a recently discovered deep sea sponge that is carnivorous. Sponges are found all over the world. The majority of sponges live in saltwater, but there are freshwater sponges too. As you can see inside the red circle, there are a lot of sponge species in the Gulf of Mexico, Florida, and the Caribbean. And there are also a lot of species in the North Atlantic, Mediterranean, and elsewhere. Virtually every time there's a scientific dive to the deepest depths of the ocean in ROVs or submersibles, a new sponge species is discovered. There are 20 sponges listed in the Endangered Species Act in the Mediterranean and northeastern Atlantic. A lot of people do not know that sponges are actually animals. As a matter of fact, sponges are arguably the oldest animals on Earth. A headline from Science Daily in 2017 reads, "New research has resolved evolutionary biology's most heated debate, revealing it is the morphologically simple sponges rather than the anatomically complex comb jellies which represent the oldest lineage of living animals." Despite the headline, I'm certain this debate is not solved and will continue into the future. On the bottom right is a photo of a comb jelly for comparison. Sponges are animals that belong to the phylum Porifera. Sponges can be found from the intertidal zone, which is the zone between high tide and low tide, to the abyssal zone. The abyssal zone is the deepest zone in the ocean, and it is in total darkness. The sponge skeleton is made of microscopic spicules that are made of calcium or silica. There are only 3 classes of sponges. Class Calcarea are the calcareous sponges, where the spicules are made of calcium. Class Hexatinellida are the glass sponges, where the spicules are made of silica. By far the largest class is the Demospongiae. 83% of sponges are demosponges made of silica and spongin. Spongin is similar to collagen, which is a type of connective tissue similar to the collagen in human bodies. All of the bath sponges have high amounts of spongin that keeps the bath sponge soft and springy and holds them together after they have been harvested and dried. Worldwide, there are only about 2 dozen commercial sponge species. Sponges are multicellular and have specialized cells, but they do not have organs. All sponges are filter feeders and bring water into the sponge chamber through thousands of pores called ostia. Water is expelled from the osculum. Sponges eat microscopic marine algae called phytoplankton, and they also eat bacteria and viruses. Many people are familiar with the high filtration rates of eastern oysters and how important they are for water quality in estuaries. Depending upon environmental conditions, an adult oyster can filter around 30 gallons of water per day, which is about 1.3 gallons per hour. In comparison, a basketball-sized loggerhead sponge can filter 304 gallons of water per hour, which is more than 10,000 gallons of water per day. Of course, an adult sponge is a lot larger than an oyster. Sponges are very important to maintain good water quality in the environment. Imagine swimming in a pool that did not have a pool filter. It would not take very long until the water quality in that pool was not that great. All sponges are very important ecologically for the ecosystem services they provide. This slide shows a photo of a loggerhead sponge on the right. Loggerhead sponges are essential habitat for juvenile spiny lobster and bonefish. In the Florida Keys, the dockside value of spiny lobster is around $45 million per year, and fishers come from all over the world to fish for bonefish. The photo on the left is a snapping shrimp. Snapping shrimp are very tiny and live inside loggerhead sponges. Snapping shrimp are the only known eusocial species that lives in the ocean. Ants and bees are the eusocial species most people are familiar with on land. These tiny snapping shrimp have a queen that reproduces and a division of labor between the worker shrimp. First of all, sponges taste terrible, and some of them don't smell too great either. The common name of one of our local species in the Florida Keys is the stinker sponge. Imagine you are a sponge. You are sessile, which means you are fixed in one place and cannot move. You may feel pretty defenseless because you don't have claws for pinching or teeth for biting. As a result, many sponges have developed chemical defenses that make them very unpalatable to predators. A lot of sponge species are high in iodine, bromine, and chlorine, which repels many animals from wanting to eat them. Sponges have very few predators, and the animals that feed upon sponges tend to be specialist feeders. For example, hawksbill turtles, angelfish, butterfly fish, cowfish, trunkfish, nudibranchs, and the Bahama star. Because of their chemical defenses, many sponges are very important for human medicines. In fact, many sponges are being studied for important pharmaceutical breakthroughs. According to Shayla, more novel bioactive compounds are obtained from members of this phylum each year than any other marine taxon. Drugs isolated from sponges are used to treat leukemia, lymphoma, lung cancer, breast cancer, pancreatic cancer, and many viruses. Drugs under current investigation live in shallow water and also in the deepest ocean depths. For more than 150 years, there has been a commercial sponge fishery in Florida. At the turn of the 19th century, sponges collected from the Florida Keys were a major commercial commodity, with export values from Key West second only to hand-rolled cigars. Tarpon Springs, often called the sponge fishery of the world, continues to be the epicenter of the sponge fishery in the United States. DuPont invented synthetic sponges in the 1940s. Since synthetic sponges were invented, the demand for natural sponges has declined substantially. At the current time, sponging in Florida is an artisanal fishery with a cultural legacy. As I mentioned before, most people are familiar with sponges that are used in the bath. In Florida, there are about 5 commercial sponge species. The most valuable sponge is the sheep's wool sponge, pictured here. Monroe County leads in landings for the sponge fishery in Florida. In Monroe County, the sponges must be harvested using a hook. There is another sponge fishery mainly in the Big Bend of Florida. In this area, the sponges must be collected by hand using a knife to cut the sponge from the base. Sponges that are cut survive and grow about 70% of the time. Sponges that are hooked survive and regrow about 40% of the time. Survival is higher when a sponge is cut from the base compared to hooking, but hooking is very inefficient because the water must be very shallow and very clear, so fewer sponges are cleared from any single area. Nearly all of the harvested sponges are sent to Tarpon Springs, Florida for final processing and sold worldwide. Sponges are also used in the medical industry, chemical industry, and for cosmetics, arts, and crafts. Sponges are vulnerable to many threats. A sponge disease in the 1930s killed many of the commercial species. It is unknown the reason for this die-off. Sponges continued to be threatened by many other causes. These threats include algal blooms, diseases, dredging, freshwater runoff, pollutants, poor water quality, and hurricanes. Hurricanes can smother sponges and also literally blow them onto the shore. The photo on the right is tons of dead loggerhead sponges that were washed ashore after Hurricane Irma. One of the biggest present-day threats are algal blooms. Beginning in 1991, there have been a series of algal blooms in Florida Bay that have killed more than 90% of the sponges living there. This was likely caused by a cyanobacteria called Synococcus, which causes the water to look like pea soup. Once such an extensive die-off occurs, the recovery of the sponge community can be lengthy due to their slow growth. Unfortunately, loss of the sponges causes a negative effect on the other species in Florida Bay. Sometimes this is called a cascading effect, when a decline in one species can cause an entire ecosystem to deteriorate because the system is off balance. As you can see in this figure, an increase in the Synococcus cyanobacteria leads to a decline in seagrasses and sponges, which in turn causes decline in water quality and the number of fish and crustaceans that live there. This can lead to a vicious cycle where recovery is difficult. This is a picture of the same loggerhead sponge before and after the bloom. This is the same picture of a basket sponge before and after the bloom. You can see that the sponges just die and deteriorate. In response to the sponge die-offs in Florida Bay, the Florida Fish and Wildlife Conservation Commission created a community sponge restoration program. Florida Sea Grant and the University of Florida IFAS Extension are partners in this effort. The Community Sponge Restoration Program is an example of a great collaboration between partners that also includes the Nature Conservancy, Bonefish and Tarpon Trust, Old Dominion University, and the Florida Keys Environmental Fund. Sponge restoration is still very new compared to oyster reef restoration and even coral restoration. It is very important that sponges do not disappear from these ecosystems because they are the dominant filter feeders and essential habitat for many animals. The species of sponges being restored are the shallow water sponges that are common in South Florida and the Caribbean. From the top right, these include glove sponge, brown branching sponge, basket sponge, elf sponge, fire sponge, and stinker sponge. Even though sponges are animals, the method to restore sponges is similar to how many plants are propagated. Sponges are cut into fist-sized pieces and attached to bricks or limestone boulders using zip ties. We call the newly cut sponge pieces propagules, and they are placed into the FWC permitted nurseries to heal and regrow before they are placed in the areas identified for sponge restoration. The Florida Fish and Wildlife Conservation Commission has at least 5 permitted sponge nurseries in the Florida Keys. Pictured here is a map of the sponge nurseries and restoration sites off of Marathon and Long Key in Monroe County, Florida. These nurseries currently hold more than 8 sponge species that are repositories for future restoration sites. Currently, the only sponge nurseries for sponge restoration are located in the Florida Keys. The nurseries are also a source for future cuttings, so that sponges do not have to be continually wild collected for restoration. The FWC is now recutting the sponges on the bricks for new restoration projects. This is a photo I took of some of the new propagules in the FWC nursery. These are photos of 4 species of sponge propagules attached to bricks. The sponges attach to the bricks fairly quickly and reorient their morphology to the dominant water currents. They also reskin fairly quickly from where they have been cut. We try to place the cut piece on the brick side. It depends on the location, food availability, and currents as to how quickly they regrow. Overall, we have found that in locations with good flow, the sponges perform very well and become reproductive within a couple of years. They overgrow the bricks into the sediment and become indistinguishable from the neighboring natural sponge communities. A on the left is a basket sponge, B is a yellow sponge, C is a glove sponge, and D is what we call the elf sponge. On the left are basket sponge propagules on bricks. These photos from Dr. Mark Butler show the growth of the basket sponges from cuttings. In less than 4 years, you cannot tell the difference between a sponge that has been restored and a natural sponge. Restoring sponge communities by transplanting cuttings of healthy sponges into areas where die-offs occurred can be an essential tool to jumpstart natural sponge recruitment. This can lead to reestablishing vital ecosystem services that sponges provide. Current sponge restoration research will examine whether transplanted sponge cuttings can restore critical ecological functions that are provided by healthy sponge communities. Raising stakeholder awareness about the ecosystem services sponges provide is critical, as they are the dominant filter feeders in many Florida coastal ecosystems, an essential habitat for spiny lobster and a host of other animals, including sea turtles. Much of this information was derived from the new Florida Master Naturalist Special Topics course called Marine Habitat Restoration. Please look for future course offerings if you'd like to learn more about sponge restoration. After you take the poll, I'm very happy to take and answer questions.